Hello everyone, this is Dr. K. Srinivas Reddy, Assistant Professor of Botany, Government Degree College for Women, Nalgonda. In my previous video, I have explained about the nucleus, structure of nucleus and functions of nucleus. In this video, I am going to explain the ultrastructure of nuclear pore complex, that is NPC. Nuclear pore complex NPC is the subtopic of nucleus, which is the part of your fifth paper, that is cell biology and genetics of your fifth semester. Let us come into the topic nuclear pore complex or NPC. The nuclear envelope is perforated by many apertures, like I said earlier in my previous video. These apertures are called as nuclear pores or nuclear pore complexes. The nuclear envelope is perforated by many apertures. These apertures are called nuclear pores or nuclear pore complexes. Here you can see in the picture very clearly this is the aperture or pore, nuclear pore. These are the pores, nuclear pores. Nuclear pores involved in the transportation of substances from nucleoplasm to cytosol, or vice versa. Means some from cytosol to nucleoplasm or into the nucleus. The function of the nuclear pore or pore complex is transportation of substances from nucleus to cytosol or cytosol to nucleus. Nuclear pores were first demonstrated by Callan and Tomlin in the year 1950. Nuclear force was first demonstrated by Callan and Tomlin in the year 1950. These two scientists studied the nuclear envelopes of Xenopus laevis oocytes under electron microscope for the first time. Callan and Tomlin studied the nuclear envelope of Xenopus laevis oocytes under electron microscope. Xenopus laevis is an African frog, which is an amphibian. In animal cells, each pore complex has a molecular mass of 125 million Daltons. Here you can see. The pore complex of animal cell has a molecular mass of 125 million Daltons, and each pore complex is composed of more than 50 different types of proteins. Each pore complex is made up of 50 different types of proteins. The proteins which are present in the pore complex are known as nucleoproteins. Nucleoporins. Nucleoporins are the proteins present in the nuclear pore complex. These proteins are arranged in octagonal symmetry. The nucleoporins are arranged in octagonal symmetry in the nuclear pore complex. Coming to the number of pore complexes. The number of pore complexes in the nuclear envelope may vary from 100 to 5 into 10 to the power of 7. There is great variation in the number of nuclear pore complexes in the nuclear envelope. The increased number of pore complexes on the envelope is an indication of active nucleus. Increased number of pore complexes on the envelope is an indication of active nucleus. It means the more the active nucleus is, greater number of pore complexes the envelope contains. More active nucleus possess more number of pore complexes in the envelope. The nuclear envelope of a typical mammalian cell contains 3000 to 4000 to 4000 pore complexes in the nuclear envelope. 3000 to 4000 pore complexes are present in the nuclear envelope of a typical mammalian cell. If the nucleus is actively synthesizing DNA, it needs to import 10 to the power of 7 histone molecules from the cytosol per every minute to pack the newly synthesized DNA into chromatin. A nucleus need to import 10 lakhs histone molecules from the cytosol per every minute to pack the newly made DNA into chromatin. When the DNA is packed with histone, it is form chromatin fiber. The DNA, when it is packed with histone molecules, it forms chromatin fiber, thin chromatin fiber. So here nucleus needs to import 10 to the power of 7 histone molecules from the cytosol per every minute. It is very important. So that's the reason why the nucleus need more pore complexes. Whenever the more pore complexes are present in the envelope, it is easy to import the histone proteins from the cytosol to the nucleoplasm, where the DNA synthesis is taking place. The same way, if the cell is growing rapidly, each pore complex also needs to transport about 6 newly assembled large and small ribosomal subunits per minute from the nucleus to cytosol. Ribosomes are synthesized in the nucleolus, which is present in the nucleoplasm. Ribosomes are synthesized in the nucleolus. So whenever the cell is growing rapidly, it needs more protein. Ribosomes are the sites for the protein synthesis. So these ribosomes are synthesized inside the nucleus. They need to transport into the cytosol where the protein synthesis is taking place. So here pore complexes or nuclear pore complexes are needs to transport the ribosomes which are synthesized in the nucleolus, which is very important, right? So here pore complexes play an important role in the transportation of the ribosomal subunits from nucleus to cytosol where protein synthesis is taking place. Each pore complex contains one or more channels through which small water-soluble molecules can passively diffuse. Small molecules less than 5000 Daltons or less can diffuse fastly through these pores. A protein of 17,000 Daltons take 2 minutes to equilibrate between cytosol and nucleus or nucleoplasm, whereas protein larger than 16,000 Daltons are hardly able to enter the nucleus at all. But how does the nucleus export newly made ribosomal subunits of 30 nanometers or import large size molecules such as DNA and RNA polymerases which have weights of 10 lakhs to 20,000 Daltons through 9 nanometers diameter and 15 nanometers long channels of pore complexes. The answer to this is that these molecules or these proteins and RNA molecules bind specific receptor proteins that can ferry them actively through these channels of nuclear pore complexes. Receptor proteins can carry these large protein molecules and RNA molecules very actively through these pores. So in this way, pore complexes are involved in the transportation of substances from cytosol to nucleoplasm as well as nucleoplasm to cytosol. Okay, yeah. Let us discuss about the nuclear pore complex or nuclear pore. Nuclear pore are circular in surface view. Nuclear pores appear in circular manner in the surface view. Okay, here you can see in the picture. This is the surface view of the nuclear pore, which appears in circular manner. It looks like a cartwheel, okay? And you remember one thing here in this picture, this is the outer membrane of the nuclear envelope, and this is the inner membrane of the nuclear envelope, okay? Inner membrane of the nuclear envelope, outer membrane of the nuclear envelope. The space in between these two membranes are called perinuclear space. The intermembrane space, which is present in between outer and inner membranes, are called perinuclear space, like I said earlier in my previous video. This is the outer membrane and this one is the inner membrane. Okay. NPC, a nuclear pore complex, has a diameter between 10 to 125 nanometers and has thickness about 50 nanometers. The diameter is in between 10 to 125 nanometers. The diameter of the nuclear pore is in between 10 to 125 nanometers and the thickness from here to here. The thickness is about 50 nanometers in thickness. Previously, it was thought a diaphragm extends across the pore. A diaphragm which extends across each pore. This diaphragm was made up of amorphous substances to fibrillar substances. This diaphragm limiting the free transfer of materials. Such a diaphragm is called annulus. It has been observed in animal cells but lacking in plant cells. Previously, it was thought that a diaphragm extends across the pore, which limits the free transfer of materials. Such a diaphragm is called annulus. The annulus along with pore form a pore complex. The annulus along with a pore form a pore complex. Let us discuss about the model which was proposed by Unwin and Mulligan. According to Unwin and Mulligan, pore complex consists of two rings at its periphery. Here you can see in the picture, this is one ring which is present towards outside, and this is another annulus or ring which is present towards inner side, or you can say nucleoplasmic side. This is the ring which is present towards cytoplasmic side, and this is the ring which is present towards nucleoplasmic side, and each ring with an inside diameter of 80 nanometers. The inner diameter of each annulus or ring is about 80 nanometers. And a large center particle, a large particle is present in the center of the pore, which forms a central plug or central granule. Here you can see in the picture, this is the central granule or central plug, you can say. A large particle which is present in the center and form a central granule or central plug. 8 radial spokes are extending from central plug to the ring. 8 radial spokes, here you can see 1, 2, 3, 4, 5, 6, 7, 8. 8 spokes are extending radially from the central plug to the annular rings, peripheral rings. As a result, the pore looks like a cart wheel due to the presence of 8 radially extending rings from central plug to peripheral annular rings. It looks like cart wheel. Okay. Sometimes particles may be attached to the cytoplasmic side of the ring. Here in the picture, particles are not present. These particles, if particles are present towards the cytoplasmic side, they are arranged octagonally. The particles which are present towards the cytoplasmic sides are arranged octagonally. The hole in the center, a hole is present in the center of the pore complex, which acts as aqueous channel through which water soluble substances shuttle between cytoplasm and the nucleoplasm, shuttle between nucleus and the cytoplasm. According to Unwin and Mulligan, each nuclear pore consists of 2 rings at its periphery. One is towards outside or towards cytoplasmic side, and another one is towards the nucleoplasmic side. And each ring has an inner diameter of 80 nanometers. A large center particle is present in the center of the pore, and which forms a central granule or central plug. 8 spokes are radially extending from central granule to annulus or peripheral rings. 8 spokes are radially extending from central granule to peripheral rings. There are sometimes particles may be present towards the cytoplasmic side, which are arranged octagonally. Here in this picture, the particles are not present. Okay, if present, they are arranged octagonally. Okay, a hole is present in the center of the pore, which acts as aqueous channel through which water soluble substances shuttle between nucleus and cytoplasm or cytosol. So this one is the old model which was proposed by Unwin and Mulligan. Okay, this model was proposed in the year 1982. Let us discuss about the advanced model of nuclear pore complex. During 1990s, significant progress has been made towards better understanding of the structure and functions of nuclear pore. New pore proteins have been identified, cloned, mutants isolated, and a detailed mechanism of cytoplasmic transfer of substances has been proposed. According to this model, nuclear pore consists of 4 separate elements. They are number 1, scaffold, number 2, transporter, number 3, cytoplasmic filaments, and number 4, nuclear basket. Nuclear pore consists of 4 separate elements. They are scaffold, transporter, cytoplasmic filaments, and nuclear basket. Scaffold, which included majority of the pore. Scaffold, which forms the majority of the pore. Transporter is the central hub which carries out active transport of proteins and RNAs. Transporter is the central hub which carries out the active transport of proteins and RNAs. Short, thick filaments attached to the cytoplasmic site of the pore or the cytoplasmic filaments. And nuclear basket is attached to the nucleoplasmic side of the pore. Here you can see in the picture these three annuli or rings form scaffold, and this is the central transporter, and these are the cytoplasmic filaments, and this is the basket or nuclear basket which is present towards the nucleoplasmic site. These are the four separate elements present in the nuclear pore complexes. Scaffold, which occupies the major part of the pore complex, and transporter is the central hub which carries out the active transport of proteins and RNAs, and cytoplasmic filaments are present towards cytoplasmic side of the pore, and nuclear basket which is present towards the nucleoplasm side. Okay, let us coming to the scaffold, which is the first element of the nuclear pore, which forms the major portion of the nuclear pores, like I said earlier. Scaffold consists of three closely arranged rings. Scaffold consists of three closely arranged rings. Here you can see in the picture, this is the ring. The ring which is present towards the cytoplasmic side is called cytoplasmic ring, and the ring which is present towards the inner side or towards the nucleoplasm side is called nuclear ring or nucleoplasmic ring, and one more ring is present in between these two rings. This ring is called central ring. So in this way, three rings are present in the scaffold. Scaffold is made up of three closely arranged rings. The ring which is present towards the cytoplasmic side is called cytoplasmic ring, and the ring which is present towards the nucleoplasm side is called nucleoplasmic ring. In between these two rings, one more ring is present which is known as central ring. The spokes of the central ring is attached to central transporter on inner side, and also attached to cytoplasmic ring and nucleoplasmic ring on outer side. The spokes of the central ring are attached to the transporter on inner side and also attached to cytoplasmic ring and nucleoplasmic ring on outside. Here you can see these are the spokes. These are the spokes of central ring. The spokes of the central ring attached to transporter on inner side and attached to cytoplasmic ring and nucleoplasmic ring to on outside. And channels are present in between. The gaps are present in between the spokes are called channels. These channels are 9 nanometers wide. The gaps which are present in between the spokes are called channels. These channels are 9 nanometers wide, and these channels allow the transportation of proteins in between cytoplasm and nucleoplasm, in between cytoplasm and nucleoplasm. Okay. Coming to the transporter, second element of the nuclear pore complex, it is the central hub which carries out active transport of proteins and RNAs. Right? This transporter is a proteinaceous ring and measures about 36 to 38 nanometers in diameter. Transporter is a proteinaceous ring and which measures 36 to 38 nanometers in diameter, and it consists of two irises which are arranged one above the other. The transporter consists of two irises and each iris has 8 arms. Each iris consists of 8 arms. Transporter is made up of two irises and each iris consists of 8 arms. And these two irises of the transporter are arranged one above the other. One above the other and moved sequentially like this. Which are arranged one above the other and moved sequentially like this. To let the nuclear proteins, to let a nuclear protein or RNA pass through the nucleus to the cytoplasm. Transporter is a proteinaceous ring which measures 36 to 38 nanometers in diameter, and it is made up of two irises. Each iris consists of 8 rings and these two irises are arranged one above the other like this and moved sequentially like a diaphragm of camera. Two irises of transporter arranged one above the other and moved sequentially like the diaphragm of the camera to let the nuclear proteins and RNAs pass through from the nucleus to the cytoplasm. Okay. In this way, transporter acts as central hub and actively involved in the transportation of proteins and RNAs. Coming to the cytoplasmic filaments, these are the thick filaments which are present towards the cytoplasmic side of the ring or nuclear pore. Okay, and the last element is that a nuclear basket, a large basket like structure which is present towards the nucleoplasmic side of the pore. Basket consists of 8 filaments and which measures about 100 meters long. In the picture here you can see only 4 filaments as it is the LS of linear section of the pore complex, so that we can see only 4 filaments and which are measures about 100 nanometers long. These elements are extending in between the nucleoplasmic ring of the pore and a small pore which is present within the nucleus and which measures about 60 nanometers in diameter. 60 nanometers in diameter. This whole structure is called nuclear basket. Nuclear basket plays an important role in the transportation of RNA or export of RNA. In this way, nuclear pore complex has 4 different or 4 separate elements, namely scaffold, transporter, cytoplasmic filaments, and nuclear basket. Scaffold consists of major portion of the nuclear pore and which is made up of 3 closely arranged rings, namely cytoplasmic ring, central ring, and nuclear ring, nucleoplasmic ring. Cytoplasmic ring is present towards cytoplasmic side and the nucleoplasmic ring is present towards the nucleoplasm side, and in between these two rings a central ring is present. The spokes of the central ring are attached to central transporter on inner side and attached to the cytoplasmic and nucleoplasmic rings on outside. Coming to the transporter, transporter is the central hub which involved in the transportation of proteins and RNA and which is a proteinaceous ring and measures about 36 to 38 nanometers in diameter, and it consists of 2 irises which are arranged one above other and they move sequentially like the diaphragm of a camera to let the nuclear proteins and RNA pass through the nucleus to the cytoplasm. And cytoplasmic filaments are the thick filaments which are present towards the cytoplasmic side of the nuclear pore, and they are attached to the cytoplasmic ring. And coming to the nuclear basket, which is present toward the nucleoplasmic side and consists of 8 long filaments, which are measures. Each filament measures about 100 nanometers long. They are extending in between nucleoplasmic ring and the smaller ring which is present within the nucleus. And the smaller ring measures about 60 nanometers in diameter. Okay, and this nuclear basket is involved in the transportation of RNA, involved in the transportation of RNA and proteins from nucleus to the cytoplasm. Okay, let us discuss about the functions of nuclear pore complexes. Nuclear pore complexes involved in nucleocytoplasmic transport of substances. Nucleocytoplasmic transport means the transportation of substances in between nucleoplasm and cytoplasm. And this transportation is mediated through a number of proteins which are present in the nuclear pore complex. These proteins are called nucleoporins or NUPs. Nuclear pore complexes involved in the nucleocytoplasmic transportation of substances. This transportation is mediated by a number of proteins. These proteins are called NUPs. A nuclear pore complex has a passive diffusion channel. Through this channel, small sized substances or water soluble substances can diffuse passively without using energy. Okay, and nuclear pore complexes can also diffuse many substances by active process where energy is used, or signal sequence mediated by carrier molecules. Here, as I said earlier, large molecules like proteins and RNA first binds with the receptor proteins which are present in the protein complexes. These receptor proteins ferry them actively through the pores. Receptor proteins carry these large molecules actively through the pores. Here proteins or nuclear proteins import through nuclear pore involves the formation of Annulus protein importing complex. Here nuclear proteins binds with the carrier proteins. As a result, a complex is formed that is Annulus protein importing complex. Here NLS means nuclear localization sequences. Nuclear localization sequences. In this way, nuclear proteins transported through the pores or nuclear pores. And export of RNAs from nucleus across the pore is mediated through NES REV protein, which is a receptor protein meant for RNA transportation. Here NES means nuclear export sequence. Nuclear export sequence, NES. NES REV protein is the receptor protein which meant for the transportation of RNA. RNA binds to this particular receptor protein. This receptor protein ferry the RNA actively through the nuclear pore. Okay. Export followed by re-import. Export of substances followed by re-import of 5S rRNA. 5S ribosomal RNA occurs through the nuclear pore by the protein with NES-like substances. Import of 5S RNA occurs through the nuclear pore by the protein, which is the receptor protein, which is like NES sequence. So in this way, nuclear pore complexes are involved in the transportation of materials from nucleoplasm to cytoplasm or vice versa. Okay, with this I conclude my presentation here. and which is very important for your academic examinations as well as competitive examinations. And I wish you all the best for your examinations. Stay home, stay safe, and we will meet in my next video. Thank you, Ananda. Bye bye.